afternoon here. Today's lecture we are going to discuss gluconeogenesis and metabolic regulation. And at the end of this lecture, we would expect you to have understood into details the following. One is the process of gluconeogenesis. Then I explain the different bypasses. Of course, we'll get into the details of those bypasses as we go through in the lecture. And you'll be able to explain the significance of gluconeogenesis. Why is this process important in your body? Then differentiate between gluconeogenesis and glycolysis, because this pathway are the exact opposite of each other. So by the end of this lecture, we'll be able to appreciate that gluconeogenesis is different from glycolysis and they are the exact opposite of each other. And then you'll be able to explain also the regulation of this pathway. And first to begin with an introduction to what gluconeogenesis is. So gluconeogenesis is a metabolic pathway that results in the generation of glucose from non-carbohydrate substrates such as lactate, glycerol, or glucogenic amino acids, and many more. So when we say glucogenic amino acids, we have some of those amino acids that can be channeled into the into this biochemical process and they yield energy. And then we have the glycerols, which is also one component from fat. When you combine a fatty acid and glycerol, that's why you form your fat. So that molecule glycerol can also be used in glycolysis to yield energy or glucose. Lactate too, which is formed during anaerobic respiration in your muscles by oxidation of pyruvate to lactate, can also be channeled into this pathway to produce glucose. Now, gluconeogenesis is one of the main mechanisms humans and many other animals use. So they keep their blood glucose levels from dropping too low. That's a condition called hypoglycemia. There are instances where your blood glucose levels get so low, and your body will need to adapt and generate more glucose from your body. That's how you end up using your protein sources, your fat sources, and then generate glucose for yourself so that your body can be sustained with some energy. So if no carbohydrates, what you're seeing CHO, I meant carbohydrates is eaten. So if no carbohydrate is eaten for a few hours, blood glucose levels will fall definitely. And then this will trigger your pancreas to produce the hormone glucagon. And this hormone glucagon is an antagonist to insulin, and it signals your liver cells to break down the glycogen stores. But then what happens when you still continue and maybe fasting, starving, or something of that kind, and you haven't taken in any source of energy? These glycogen stores can get done, and glucagon presence in your blood or in your body would also result into stimulation of this process called gluconeogenesis. Your body will need to tap into other sources of energy, which we call the secondary sources of energy. The primary sources are basically carbohydrates. And then when you look into where this process occurs, this gluconeogenesis occurs majorly in the liver and the kidney cells. And this process as well require energy for it to take place. So glucose mainly formed from 3 carbon molecules, that is a pyruvate, that is during our glycolysis. And this pyruvate is also formed from glucogenic amino acids. AAS means amino acids. These amino acids can undergo deamination, and then they end up being channeled into the glycolytic pathway and produce the pyruvate. And other major gluconeogenic substrates include lactate and glycerol. And this process can also be stimulated by the hormone epinephrine, and this hormone response helps your body respond to emergencies. Look at an instance where you're just moving and then all of a sudden you're scared. Maybe you have a snake in front of you. Your body will automatically need to generate some energy for that. In that instance, such shows that there's a response to that, that your body will need to generate more energy for you to do that. Even when you're feeling so weak before, but when you're in that emergencies, you really need your body will really have to trigger production of such energy. Gluconeogenesis is essentially the reverse of glycolysis, like I said before. You can simply say gluconeogenesis is the opposite of glycolysis because glycolysis is breaking down of glucose to pyruvate, and gluconeogenesis does the reverse. From pyruvate, takes it up direct to glucose, which your body can still use. However, three of these reactions of glycolysis have a forward direction, that they are essentially irreversible. So in this process, there are some steps that you cannot reverse. If we say glycolysis is the opposite of gluconeogenesis, that simply implies, in other words, we can just reverse those arrows. And but there are some of the steps in the process that are not reversible, meaning you have to go through in a bypass. Like I said in one of as we were talking about our learning objectives, you have to understand these bypasses, how they occur. So one of these steps is the hexokinase, where hexokinase, that is about the first step, and then phosphofructokinase, and then pyruvate kinase. Those are basically the enzymes that catalyzes the steps that are irreversible. Meaning in this way, we have to do a bypass process. The body will have to go through a bypass so that the step can continue since it is not reversible. And these steps must be bypassed in gluconeogenesis. Two of the bypass reactions involve simply hydrolysis reaction, which is the hexokinase and the phosphofructokinase. These are the two last bypass steps. And since this is a reversal, in glycolysis, pyruvate kinase is one of the last steps. But in gluconeogenesis, it becomes your first step and the first bypass. Then the second bypass is the phosphofructokinase enzyme catalyzed reaction, and then lastly hexokinase. And the last two bypasses, which is for phosphofructokinase and the hexokinase, are simply bypassed by hydrolysis reactions. But then for the first bypass, that is the pyruvate kinase, there's really a lot of kind of reactions that has to take place. And we shall get to the details later. Let me first show you. Right here is an illustration of the reverse, where you see on my left or the left of the screen with the red is what you call the glycolysis reaction. Then on the right hand side where you have the blue writings, that's where the gluconeogenesis, that's the gluconeogenesis reaction. As you can see, the arrows are reversible, and the arrows are moving in the opposite direction, the blue and the red arrows. So that's clearly explains what I said before, that glycolysis is the opposite of gluconeogenesis. And where you see the curving arrows, those are the bypasses. However, the rest of the other steps have the same enzyme, same product. So to continue with the bypasses, the first bypass, this is the conversion of pyruvate to phosphonopyruvate, which is what we call the PEP. And pyruvate being the final product of glycolysis, it has to be converted back if we are to take a reversal of this process. And this reaction cannot occur by just a mere reversal of the pyruvate kinase reactions of glycolysis. So pyruvate is first transported from your cytosol into the mitochondria, or is generated from alanine within mitochondria by transamination. And alanine is one of these glucogenic amino acids that we've been talking about earlier on, that some of these amino acids can be channeled in for this process. And how does this happen is that a transamination process occurs, in that, just like the way you're seeing in the illustration or the figure below, is that alanine is being transformed and converted into pyruvate. And this is the reaction called the alanine transamination process. And it takes place in with the presence of an enzyme called alanine aminotransferase enzyme. And conversion of pyruvate to phosphoenolpyruvate occurs in two steps. It's not a single step that converts it. And one of these is the step that involves pyruvate carboxylase. And first, pyruvate is converted to oxaloacetate. Oxaloacetate, as if you remember, our glycolysis, it's one of the intermediates with four carbons. So this reaction it requires an enzyme called biotin. And this biotin is a carrier of an activated bicarbonate, or we can say hydrogen carbonate. Above is a reaction that shows this that a pyruvate that has been reacted with the hydrogen carbonate gives us oxaloacetate ADP, and that this reaction utilizes energy for you to burn this to add a carbon from the hydrogen carbonate to pyruvate. We know pyruvate is a two carbon molecule, and oxaloacetate uh, is a three carbon molecule. So for you to add this to form the pyruvate, you will need to add in more of this into the reaction. So the bicarbonate here reacts with pyruvate to form the oxaloacetate here. And this mitochondrial membrane do not have oxaloacetate. And so with that, before export to the cytosol, the oxaloacetate formed from pyruvate must be reduced to malate. Just like I've explained, I've said earlier here that the mitochondrial membrane do not have transporters for oxaloacetate. So this oxaloacetate cannot get out of the mitochondrial matrix into the cytosol, where the rest of the process or conversion glucose can continue from. So for this matter, we'll have to convert this oxaloacetate into malate. And what happens here is that malate, the malate later on, after being converted or after being produced in the matrix, will be transported now into the cytosol and then converted back into oxaloacetate. This is basically the reverse. This is basically the reverse of everything that is happening here. In a way that you're seeing oxaloacetate is being converted to malate using the NADH. And then and then and then this reaction is catalyzed by the dehydrogenase. So the same enzyme still catalyze the reversal reaction, where the malate here is converted back into oxaloacetate. So the sole purpose of converting oxaloacetate in the matrix to malate is to allow it to be transported out of the mitochondrial matrix into our cytosol. Upon transportation out into the cytosol, and it will be converted back into the oxaloacetate, and this reaction, like I said earlier on, it's been catalyzed by a dehydrogenase enzyme. And in the biochemical processes, once you see the NADH and NAD plus or FADH, NADPH, all those ones are catalyzed by dehydrogenase enzymes. So you just have to take note of that. The oxaloacetate here, after being transported to into the cytosol, is then converted into PEP. PEP here is the phosphoenol pyruvate, one of the intermediate. in glycolysis, which and this is the second last intermediate that is converted into pyruvate. So what happened is that the oxaloacetate here is converted into PEP by the action of what you call the PEP carboxykinase enzyme. And this uses GTP. GTP is one of those molecules that provides energy, just like ATP. They have literally the same function, so they perform actually the same thing. What happens here is that oxaloacetate is converted to PEB, phosphoenolpyruvate, with release of carbon dioxide from here, and then a phosphoryl group is donated from the GTP to our phosphoenolpyruvate. And the second bypass right here and is the one that occurs in step 3, where we have the fructose. Step 3 of glycolysis, where we have the fructose 6-phosphate, and using ATP is converted to fructose 1,6-bisphosphate as a phosphorylation reaction. And we said earlier where there is a phosphofructokinase enzyme, and that is exactly the step number 3 in glycolysis. But then to reverse this, and I think some of you may be asking, how did you reach to this step now? The rest of the step after formation of PEP is basically the same thing, and it's just the reversal thing. Just like we have talked earlier, we said here you form pyruvate. I think hopefully you're following my cursor closely. On the blue side, we have pyruvate into oxaloacetate and forming. From oxaloacetate, we have PEP carboxykinase forming our phosphoenolpyruvate. So the rest of these steps up to step number 3 of glycolysis are really the same steps and the same enzyme, meaning phosphoenolpyruvate, which is the PEP, can be converted to the phosphoglycerate catalyzed by enolase enzyme, and then later on it is also converted to 3-phosphoglycerate by this very enzyme as of glycolysis called phosphoglycerate mutase, and then again converted to 1,2-bisphosphoglycerate by phosphoglycerate kinase. Then from there, that same intermediate, the 1,2-bisphosphoglycerate, is converted to glyceraldehyde 3-phosphate. And then finally, same reaction still happens, and then where our dihydroxyacetone is converted to our fructose 1,6-bisphosphate. So after formation of that, now that's where the bypass comes in, our second bypass that I was on. Hopefully we are not lost. Our second bypass, which is right here, that fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and this is where we have the phosphofructokinase enzyme in glycolysis. Now in gluconeogenesis, this step is irreversible, just like the last step that I talked about, its bypass. In this, like earlier on, I discussed saying 2 of these steps, which is the step 3 of glycolysis and step 1, are just reversed using hydrolysis reaction, just simple addition of water, and then you're done with reversing it. So in this case, you're going to add water to fructose 1,6-bisphosphate, and then you form the fructose 6-phosphate and an inorganic phosphate. And the reaction is catalyzed by a different enzyme. It's not phosphofructokinase, but in this case, the enzyme is called fructose 1,6-bisphosphatase. So you have to note this, that because this reaction is highly spontaneous and therefore irreversible in intact cells, the generation of fructose phosphate from fructose 1,6 bisphosphate is catalyzed by different enzyme. It is magnesium dependent fructose 1,6 bisphosphatase. So this enzyme really requires magnesium ions to function essentially or perfectly. And from here, this is an illustration of the second bypass that we have fructose 1,6 fructose 6 phosphate being converted to fructose 1,6 bisphosphate. So that's just an illustration or a visual to make you better understand the process and the bypass, the second bypass. The third bypass and the final reaction is that this dephosphorylation of glucose 6 phosphate to yield glucose. Remember, in glycolysis, the first step was to phosphorylate glucose to form the glucose 6 phosphate. In this reaction, you have to remove the phosphate group. Dephosphorylation now, meaning we are removing the phosphate group. And that initial reaction in glycolysis is catalyzed by hexokinase or glucokinase. But then for gluconeogenesis, it is a different enzyme, and the reaction here is just like we said earlier on, by just simple addition of water, you form your free glucose and a phosphate group that has been added before. And this reaction is catalyzed by glucose 6 phosphatase. A simple way to remember these reversal enzymes is that we have phosphatase for the reverse reaction, where we have glucose 6 phosphatase. We have in the third is the fructose 1,6 bisphosphatase. So you just add the test reaction to its product. So that is basically what it is. Those are the reversal reactions that take place. And this enzyme called glucose 6 phosphatase is embedded in the endoplasmic reticulum (ER) membrane in the liver cells. And here is the illustration, a visual to show you the reaction. And then biosynthesis of carbohydrates. In essence, gluconeogenesis is reverse glycolysis apart from glycolytic steps that must be bypassed, just like we have discussed earlier on. That gluconeogenesis is the exact opposite of glycolysis apart from the three steps that we need to bypass. So that's basically it. The importance of gluconeogenesis here is that a continuous supply of glucose is necessary as a source of energy, especially for the brain, the nervous system, and the erythrocytes. So just in case your body stores get down, the energy stores are really low, glucose levels have gone low, and your body can tap into or can supply energy for all these body systems, such as the brain, the nervous system, the erythrocytes. All these can be provided for from this process called gluconeogenesis. And gluconeogenesis is used to clear the products of metabolism of other tissues from the blood. Example, lactate. During anaerobic respiration, if you form lactate, what else do you do with it? Your body can't use it, and lactate also have a tendency of causing you muscle cramps. That's one of the effects. Your body system can get rid of this, and in a way that it is utilized by the body, by the body it is utilized by the body for energy, which is very important. You get rid of lactate and also provide energy for your muscles. Another thing is erythrocytes. All these ones can be broken down. Erythrocytes, these are the red blood cells. They can be broken down. They are protein in nature, and then you can get energy from them using the amino acids that can be gotten. Glycerol is a byproduct of breakdown of these fats, and that also can be that. There is alanine cycle. That's one of the mechanisms through which your body can get rid of excess amino acids in your body while replenishing your energy stores. So it can convert alanine. It can convert the amino acids into energy, simply glucose. So you get rid of this metabolic waste. The body doesn't have stores for amino acids. It just have amino acid pool. Your body cannot store those amino acids. So they get rid of it in a way that it also generates for you energy. And that's one of the importance of gluconeogenesis. Without it, there is possibly no way your body can get rid of those amino acids easily without causing harm to you. And during starvation, gluconeogenesis here maintains the blood glucose levels. Yeah, just like we have understood the process, glucose levels can be maintained through gluconeogenesis because it's a process that generates energy or generate glucose when your body is depleted or the carbohydrate stores of glucose are depleted. Then regulation of this mechanism is that during starvation, stored glycogen is typically used up within the first 10 to 8 hours. So what happens? Gluconeogenesis and glycolysis are reciprocally regulated. This means the reverse regulation. Whatever stimulates glycolysis inhibit gluconeogenesis, and whatever inhibits glycolysis will stimulate what? Gluconeogenesis. And one pathway is relatively inactive as the other is active, so meaning the two cannot coexist at the same time. As one is activated, the other goes down. So this is a regulation by enzymes, and there is also regulation by hormones and ATP. We understand ATP as adenosine triphosphate. There is an allosteric regulation that we have that starting substrate for gluconeogenesis is oxaloacetate. So it's transported across mitochondrial membrane into the cytoplasm as malate and then reconverted to oxaloacetate by reduction of malate by the NAD. What does this mean? Allosteric regulation, if we get to look into enzymology, it's more like a substrate inhibiting the process that forms it. So in this case, if there is a lot of oxaloacetate in the body, it will inhibit the process. That's how it regulates this. If it will inhibit glycolysis, and gluconeogenesis will proceed as fast as possible, so that this concentration of oxaloacetate is reduced from the body. And here are other enzymatic or allosteric regulations that we have. And where you're seeing pyruvate carboxylase here is inhibited by PEP carboxykinase. Fructose 1,6-phosphatase and glucose 6-phosphatase. So the presence of these enzymes can also cause an effect on that, on the regulation. Above is the table that can indicate all this. Formation of oxaloacetate can be inhibited by glucose and fructose 6-phosphate and the PEP phosphoenopyruvate. So that's basically is an instantaneous process. It just occurs instantly and is a very fast one, no time lapse that take place. So it can interswitch itself so easily and very fast in the process. Difference between hexokinase and glucokinase, like we said earlier on. Hexokinase dominates in low glucose states. Let's first understand where this thing comes in from. In the process of glycolysis, we have these two enzymes, hexokinase and glucokinase. And then we see them at what instant that one enzyme take over the process over the other. We have that glucokinase here dominates at higher glucose concentrations and is present in liver and absent from muscles. Meaning glycolysis taking place in the muscles is really catalyzed by hexokinase. And that in the liver is catalyzed by glucokinase. So hopefully this is the difference between the two enzymes is clear. This wasn't really important for us to bring. This would have come in glycolysis, but I love to at least put something here and emphasize on it, just to help you add on what you learn from glycolysis. Going further, still under the regulation, is that we have hormonal regulation of gluconeogenesis. In that glucagon and glucocorticoids increase gluconeogenesis. And to understand this is that glucagon is one of those hormones produced from the pancreas, and glucocorticoids are also some of those substances that can result into an increase in gluconeogenesis. Meaning, in presence of glucagon and glucocorticoids, the process of gluconeogenesis is increased, or there's a speed in its process. There's a faster reaction in the process. Then insulin, which is an antagonist to glucagon, inhibits this process. So meaning when glucagon levels rises in your body, the cellular AMP, adenosine monophosphate levels also increases. So adenosine monophosphates, these are some molecules in your body. Uh, and then there's the CAMP, which is the cellular adenosine monophosphate concentration, inactivates the enzyme pyruvate dehydrogenase. So the connection here is that when when glucagon levels rises in your body, it results into an increased CMP levels in your body. But what does this increase do? It results into pyruvate dehydrogenase inactivation. Once this enzyme is inactivated, and then the process of gluconeogenesis will increase, meaning it's more like a dysphosphorylation of the substrate that are happening there. Because during glycolysis, we have been phosphorylating all these, most of these intermediates, adding phosphate groups. And in this reaction, when the CMP concentration increases, then the dysphosphorylation process is activated. And glucagon also stimulates the production of phosphoenolpyruvate carboxykinase, that is a PEP kinase, and a fructose 1,6-bisphosphatase. Can do these enzymes in terms of regulations. These are some of the key enzymes involved in gluconeogenesis, especially in the bypasses. PEP carboxykinase in the third bypass, the fructose 1,6-bisphosphatase phosphatase is involved in the second bypass. So this ensures that more pyruvate is converted to oxaloacetate and thereby channeled into gluconeogenesis. Ethanol here can inhibit this pathway. So ethanol is formed during fermentation, or if pyruvate is fermented, it forms ethanol. That pathway we have discussed it earlier in one of those reactions. And then insulin is one of the hormones also that say glucagon stimulates production of phosphocarboxyl counter by inhibiting gluconeogenesis. So this hormone works in the opposite way, an antagonist, like I've mentioned it many times, that is an antagonist to glucagon, sorry. And an increase in insulin stimulates the expression of pyruvate kinase. So it's more like if you have more insulin, there is more pyruvate kinase in your body. This is pyruvate kinase, then phosphofructokinase, and these are the enzymes that makes and degrades the fructose 2,6-bisphosphatase. So these enzymes degrade the enzyme that promotes gluconeogenesis. Meaning, in presence of insulin, gluconeogenesis process is decrease and the effect of glucagon is counteracted. The hormones glucagon and epinephrine respond to low glucose levels by inhibiting glycolysis and then stimulating increased concentration of cellular adenosine monophosphate, which is the CAM. So, like we saw earlier on, we know what the role of CAMP. So, there can be a question that describe or explain the role of CMP in relation to glucose, in relation to glucagon and insulin. Or what is the effect of CAMP on the levels of blood glucose in relation to insulin and glucagon? You can be able to interrelate how the CAMP is affected by insulin levels and how they result into the rise or decrease, how they affect this biochemical process, each one of them, gluconeogenesis and glycolysis. So you can interrelate this, and that should be a synthesis question. If you get time, at least you can try to think about it and answer that. Then type 2 diabetes is marked by excess glucagon and insulin resistance. So insulin can no longer inhibit the gene expression of enzymes such as the PEP carboxykinase, which lead to increased levels of hyperglycemia in the body. What happened here? A diabetic patient is one that cannot regulate the blood sugar levels, and there is a type 2 diabetes especially is one that is associated with insulin resistance and other diseases like hypertension and obesity and uncommunicable diseases. So what happened here is in a person who is diabetic, we have seen how this is important in regulating our blood sugar levels. So in such a way that your body cannot express enzyme like PepCK, carboxykinase, meaning the process of gluconeogenesis is actually affected. So the anti-diabetic drug metformin is one that reduces blood glucose primarily through inhibition of gluconeogenesis on overcoming the failure of insulin to inhibit gluconeogenesis due to insulin resistance. So how are we going to understand this? Metformin is a diabetic drug that is used by many people who are diabetic. Like the statement here says, it inhibits the process of gluconeogenesis. Let's get to understand this. What's the relationship and what's the effect of gluconeogenesis, glycolysis on a patient who is diabetic? Glycolysis is very important and it helps a person who is diabetic with the breakdown of these glucose levels, the glucose. However, on the case of gluconeogenesis, it generates more of the glucose, and meaning it increases your blood sugar levels. So, since your body can no longer produce insulin or is resistant to insulin, there will be high levels of glucose levels in your body, because your body now, for you to have the expression and production of, to inhibit the enzyme PEP carboxykinase, which is a key enzyme in the process of gluconeogenesis, you have to be your cells should be responsive to insulin that will help in reducing levels of CAMP and also inhibiting PEP carboxykinase enzyme. But then in this case, a diabetic person cannot really go through this process because of the insulin resistance. So the cells are not responsive to insulin, and the cells really have a problem with that metabolism. So in this case, your body does not produce the enzyme or inhibit the process that produces the enzyme PEP carboxykinase. So PEP carboxykinase will continuously be produced and gluconeogenesis will continue happening, and more glucose is produced in your body. That affects the diabetic person so much. But here we are, pharmacologists, the pharmacists have come up with a drug which is anti-diabetic, and this drug is called metformin. This will reduce your blood glucose primarily through the inhibition of gluconeogenesis. This enzyme helps in inhibiting that because your body has a failure to respond to insulin. So this will help your body not to continuously produce glucose in your body through gluconeogenesis. Hopefully this is clear, and that can be a very good synthesis question if you get to understand. A summary table right here that says the enzymes activated by and inhibited by. So we have pyruvate carboxylase, which is activated by cortisol, which is a corticosteroid, a glucagon, adrenaline, acetyl-CoA, and it's inhibited by insulin and adenosine diphosphate. Then the PepsiK, which is a phosphoenolpyruvate carboxykinase, is activation has not been shown. It's inhibited by insulin. The fructose 1,6-bisphosphate, one of the intermediate, is inhibited by CAMP and fructose 2,6. So if you and then glucose 6-phosphatase, the last enzyme in gluconeogenesis, is inhibited by insulin. If you get to understand all these things, you definitely have to be able to understand and answer questions that comes around this, and most especially synthesis questions, and not a cram work. I discourage students from doing cram work and everything. In the real world, we have to focus on that. Otherwise, thank you very much for this lecture, and I say thank you for today.